Okay, thanks everyone. Hi, thanks to the organisers for giving me the opportunity to talk today. Um, oh, sorry. Sorry, I'll just start a timer to make sure I don't go too far over. So yes, thanks to the organisers for giving me the opportunity to speak here today. And I'm going to talk about some work that we've been doing now for a few years, looking into the role of the gut microbiota in vaccine responses, particularly in infants. And I know my title is very, very long, but just remember, AIR study is the name of the cohort study that I'm going to be talking about. And I also have a poster that will be tomorrow evening. So I'm going to start with the acknowledgement, actually. So I led the multi-omics kind of bioinformatics part of this, but was part of a large team under Professors David Lynn and Helen Marshall in South Australia, together with Grant Rogers as well, who helped with the microbiome work, and Professor Preeta Richmond in WA. So when I first joined David's team back in 2018, this was kind of new to me, that vaccines don't just work for everyone. And I think now the world is kind of aware of this, right? This is an example of the infant national immunization program schedule from Australia, but you'll see very similar things around the world where infants get vaccinated against a broad range of different pathogens. Pre-COVID-19, these sorts of programs were estimated to save up to 3 million lives a year, not to mention actually just preventing severe illness, which is measured separately to deaths. And we typically measure success of these vaccines through antibody responses, not for every vaccine, but for most. But these antibody responses can be really variable between individuals, up to 40-fold for Haemophilus influenzae type B, for the seasonal influenza vaccine up to 100-fold. And there's quite a good body of evidence now that there's a role for the gut microbiota in this variability. Our team published this paper back in 2018, and this was very much the prelude to what I'm going to talk about later. This was preclinical work in an antibiotic-exposed mouse model, where we basically could show that antibiotic exposure was sufficient to impair responses to a bunch of those vaccines that infants get in early life in mice. And there's plenty of other people working on this as well. Bally Pullendren's team in America is doing some really cool stuff in this area too. And there's a lot of studies that have looked at correlating composition of the gut microbiota with antibody responses to, particularly in SARS-CoV-2. And our team also reviewed this in Nature Reviews Immunology, discussing both kind of the evidence for it, as well as proposing potential mechanisms. So if it's something you're interested in, please check out that paper. And that leads into the cohort study I'm going to talk about today. So like I said, this is the AIR study, the Antibiotics and Immune Responses study. So this was a prospective observational study where we recruited about 255 infants. These were all healthy, full-term, vaginally delivered, almost all were breastfed. So generally a healthy cohort who received some sort of antibiotic exposure then in early life. We just monitored this exposure and then basically slotted these infants into a priori defined exposure groups. These exposure groups were, I have them labeled up here, and I'll use these colors throughout: the blue for no ABX, red for what we call the neonatal ABX, yellow for the intrapartum, and gray for this postnatal. The intrapartum was basically moms that, almost universally was moms that got antibiotics for group B strep prophylaxis. And this red group was babies where there was kind of suspected early onset sepsis, but then it was negative. And this is quite a common occurrence. Something like for every one case of culture positive sepsis, there's about 100 babies that will actually get antibiotics for it. And then we also had this, a third exposure group. These were babies that were breastfeeding, but mom was taking antibiotics. And these exposures were all basically within kind of birth to maybe week 2 of life, but very much in this early period. This is when these exposure groups are defined based on. We then looked at vaccine responses to several of these routine immunizations that infants get in early life. We looked at these 7 and 15 months later. So the exposure groups and the vaccine responses being measured are really quite far apart. The reason for that, of course, is to give your adaptive immune system time to actually, mount a sufficient response. And then we also slotted in some multi-omics around this, particularly around the time of vaccination, or the time at least they received the bulk of their primary immunizations. Shotgun metagenomics, bulk total RNA-seq on blood. We also had some flow cytometry in there too. And so I'm just going to give a very brief overview of what we found, and then I'm going to dig into a couple of the data sets and just show you a few snippets from that. First was obviously antibiotic exposure, particularly in that neo-ABX group, lowers gut microbiota diversity, no shocker there. We also found it lowers bifidobacterium species. It also led to what we saw in our RNA-seq, much higher levels of inflammatory gene expression, particularly related to monocytes and myeloid-derived cells. Also lower B-cell gene expression, B cells being cells that produce antibodies. So this obviously was interesting to us. And then at month 7 and month 15, really only in that neo-ABX group, babies that received direct antibiotics in the first week of life, we saw really significantly impaired responses to those scheduled vaccines. Obviously, this is an observational study, but we have a parallel preclinical arm to this as well, where we can see in germ-free mice, some vaccines, not all, are really impaired. That's the blue line at the bottom, green being just a regularly colonized SPF mouse. And then we can basically use probiotic to at least partially restore those responses in germ-free mice, where in this case, particularly interested with this probiotic is it's used in neonatal intensive care settings in Australia already. So in terms of safety and stuff, obviously, it's a bit easier to translate. Jumping over to some of the data. So this is just, we measured 20-something different vaccine antigens across 3 different time points. This is just 3 of them at month 7. 2 of them from the pneumococcal vaccine, and then the one at the end is the Haemophilus influenzae type B. And just note again on the y-axis, that's a log 10 scale. So there's really quite a significant drop in these infants. On top of that, there's actually clinically defined thresholds of seroprotection for these as well. And these infants have now been pulled below that threshold, at least on average in these groups. So really quite a strong effect in this, because again, we're talking separated by 7 months, and some of these changes actually last out to 15 months as well when we stopped monitoring. Jumping over to some of our bulk RNA-seq data. So this heat plot here shows normalized enrichment scores relative to the no-ABX group. So red is higher, blue is lower, and the stars indicate statistical significance. And what we can see is in this neo-ABX group, which is this first column here, all up here, this is basically related to monocyte expression, neutrophil expression, TLRs, that sort of thing, all much higher. This is basically the same day that they would have gotten their primary immunizations. So just resting at baseline, higher levels of inflammatory gene expression, and then lower levels of, down here, modules for B cells, plasma cells, that sort of thing. Just as an aside, if anyone is integrating a kind of bulk RNA-seq data with metagenomics, these blood transcriptional modules, they were defined in a paper, I think in Nature Immunology in 2014, and we've just found them super useful for reducing the dimensionality of your RNA-seq data down from whatever 10, 15,000 features down to something like 200, while keeping a lot of or keeping like labels that actually make sense from an immunological point of view. And then you can also use these in something like a gene set variation analysis, which is what I've done here, that will then convert your count table over to something where the features are then just your, in this case, blood transcriptional modules. This is just showing some of those impairments and some of those heightened ones there. You can see this is up, not by a lot, but I'll actually come back to this again in a second. And then these B-cell related activity of these B-cell related modules, this was directly positively correlated with vaccine responses 7 months later, whereas the inflammatory ones were directly negatively correlated with those. Of course, this was bulk RNA-seq data, and everyone will always tell you, well, it might just be changes in cell frequency. We also looked at cell frequency, and it wasn't actually. These infants really had the same level or the same number of immune cells in their blood, and we measured 40-something different populations. This is just the B-cell one, just to give you an example of what that looks like. And then I'm just going to finish by talking on some of our metagenomic data sets. So we've, I think, a total of 409 samples that we did shotgun metagenomics on in this cohort. We also measured bacterial load through a 16S qPCR. Interestingly, bacterial load, while it was associated with composition, no change across any of the antibiotic exposure groups. The antibiotic exposed infants did have lower diversity, as you'd expect. But no difference in bacterial load, which I was really advocating that we measure this, and we spent a lot of money measuring this, so I was like, oh. But we did find between week 1 and week 6 that the samples, typically the load increased and the diversity increased as well, kind of consistent with that community growing in complexity as the infants age. Jumping over to some of what we found different in differential abundance analysis between those ABX groups. This is a volcano plot at our first time point and our second time point. And then the color represents which ABX exposure group. The x-axis is fold change with the directionality increase or decrease, and then a negative log 10 transformed p-value on the y-axis. And what we can see is the intrapartum antibiotics, it doesn't impair your vaccine responses, but it does actually change a bunch about the microbiota. And actually, I probably should have mentioned that back in the RNA-seq data too, but we did see in these infants, there was actually a bunch of other changes in their NK cells and other things too that although why it might not be relevant for in this context in vaccines, does seem that intrapartum antibiotics are doing something at least. But we can see out to, this is at week 6, again, the time that they were receiving their immunizations. The infants that directly had antibiotics still have this lower level of bifidobacterium. And so then this is just some of those species just plotted, so you can see kind of what the distribution looks like. It certainly wasn't universal. There was, control infants with no exposure, with low levels of bifidobacterium, and there was exposed infants, with high levels of bifidobacterium. And this goes to sort of the personalized nature of how your microbiome will respond to antibiotics, right? This is so then moving on from that, we just looked at whether or not there was a correlation between the week 6 microbiota, and the month 7 antibody responses. And there was, at least here, just at several different taxonomic levels, different bifidobacterium species, family, genus, and so on. And like I said, we tested that with this probiotic that contains bifidobacterium, and at least in mice, we can show that there's that causal association there too. And then moving on to lastly, kind of how so the way we had registered that this is kind of a study, we very much had this primary outcome related to, oh no, my text messed up. Oh, well, anyway, we had this primary outcome related to our antibiotic exposure groups. But then kind of as a sub-analysis from that, we defined community types in these infants, so cluster infants based on their microbiota, and then look at those microbiota groups in the other omics data. And this is a relatively, it's probably the simplest way really of being able to integrate kind of various data sets. And basically what we can see from that, again, sorry for this, is there's several different bifidobacterium groups. There's this group here that's kind of a mix of Klebsiella and Streptococcus, an E. coli group. This group has more Bacteroides and so on. What we can see is this increase in the inflammatory signatures. It wasn't actually universal. It was only 3 of the community types had that, and the other 2 didn't. The other 3 didn't, I should say. And similarly with that impairment in the B cell responses, that wasn't seen universally across all the community types. So what it seems to be is that the antibiotics, will have some chance of shifting you to one of these groups where you may or may not get this immune phenotype. And yeah, with that, I will finish up. So again, thanks everyone for listening. Sorry about that last slide. And please come talk to me at my poster. Thank you.
Okay, we have time for some questions.
Hey, thank you for a really interesting talk. I was wondering if you looked at the resistome composition using the metagenomic data, and if there was any differences within the resistome composition.
Yes, there was. I didn't include it today, but come by the poster. I can actually show you the data if you want. And yeah, I guess there was changes in the resistome, but they were really strongly linked to the composition. So it didn't look like it was plasmids suddenly jumping in. It was just, you're now shifted over to gram negatives, and as a result, you have a lot more resistance genes being carried on those. But yeah, there was. We did see, particularly at that week 1 time point, the exposed infants all had this big jump in MEC-A, which is what's used to characterize MRSA. So that was quite concerning. Yeah.